So can I start? Yeah. So thank you very much for coming today. And today I will be talking about a project that I'm developing here in the department, together with some other colleagues, as Pablo Matos and some smart people abroad. So the title of my presentation is Merging Next Generation Sequence Technologies. So as you know, I'm not an ecologist, and most of the time I'm here at Kokomo listening, and I cannot get so many things. But in evolutionary biology, we are interested to study the evolutionary relationships among the organisms. So the main goal of my research is to infer phylogenies. But just having phylogenies for the groups is not enough for us to understand the whole picture of biodiversity. So we needed to couple these phylogenies with some other studies. So, for example, in evolutionary ecology, and one of the examples, two examples that we can use phylogenies in ecology, in ecological studies, is, for example, examining the phylogenetic structure of community assemblies, or, for example, exploring the evolution of traits, for example, beta and alpha. But as I said before, I'm not an ecologist, so perhaps you have much more examples how we can use phylogeny in ecological studies. And to infer the phylogeny, we can recover data to infer these studies, and we can classify in two broader categories. So one way to infer phylogenies is using molecular morphology data. So when we think about morphology, we always remember about external parts, right? We always remember the shape of the wings in some butterflies, or we always remember the leaf shape or some kind of things, but we always forget about that we can use the anatomical data and also chemistry data and convert somehow this data in morphological matrices to infer phylogenies. But nowadays, to be honest, it's not easy to see publications about this because it takes a long time to collect this data. For example, for you to have a whole picture how the morphology of a particular species is, you need to go to several herbarium and analyze thousands of materials, and this costs time and also money. And this is not the topic of this talk today. I will be focusing inferring phylogenies using molecular data. And the evolutionary studies until 2004, they were based on this type of sequencing. So we call Sanger sequencing era. So imagine that here we have three species that we will want to infer a phylogenetic hypothesis for this group. So we needed to have a DNA extraction of these samples, this species, and later we needed to select the genome that we want to infer the phylogenetic hypothesis. So we can infer phylogeny based on the nucleus data, mitochondria, or for example, in case of plants, we have a third genome, which is the chloroplast. And the second thing that we need to choose is which molecular regions we are going to use to infer this data. And these two questions, or this two selection of data, is highly correlated to what you're trying to answer. Are you trying to answer how the organisms are related in terms of family level, in terms of species level, in terms of subspecies level? So depending on your questions, you have a genome that is more appropriate and also a molecular region. And after this, you need to sequence every single region that you are doing. And all of these experiments cannot be done together. So you need to extract, you need to amplify one region one by one, and also sequencing this one by one. So you may realize how much time does it take. And this is just example of 3 species and 4 molecular regions. But after 2014, a new revolution in the sequencing data comes. And this is what we call next generation sequencing era. So to infer the phylogeny for the groups, you still need to extract the DNA, but the selection of the region could be from one molecular region to whole genome sequence at the same time. So you don't need to do experiment one by one. And the sequencing of these regions that you're going to select can also be done at the same time. So this is much more faster compared to center sequence technology. So this is just an example. For example, the Human Genome Project was published in 2003. And in the human genome, we have 50,000 genes. And it spent 13 years analyzing and getting this data. And it's costed $3 billion and involved more than 2,000 researchers and 20 institutions. So it took a long time. And nowadays, with the next generation sequencing, you can have the human genome sequence in 1 or 3 days. So this is a very huge advance for science. And it costs only $1,000. So it's huge also in the price. But the problem is that to analyze next generation sequencing data, you need to have some expertise in bioinformatics. And bioinformatics is not easy, and it makes people cry. Just because we are not here mostly describing genomes, right? We are interested in the relationship of the organisms. So in evolutionary biology, I'm going to show you how much time does it take to collect this data. So during my master's, I used 3 molecular regions, 24 species, and I took 1.5 years to collect this data. But in my PhD, this publication is out today. If you want to check, I recovered 942 regions and 111 chloroplast regions for 64 species, and I took the same amount of time. So when I say things about the next generation sequencing, it seems so easy, right? Everyone wants to do next generation sequencing, but doing that is not so easy. First, you need to choose the sequencing can be classified in 3 types of generation. So the center sequence generation, where you need to amplify or select region by region, is called the first generation sequencing. And it's high accurate, but involves a lot of lab work. And I was saying before, you need to select region by region, and it's very expensive, as I showed in the human genome example. And the data output that you have is from 300 into 1,000 base pair, usually. And the next generation sequencing, the second generation, involves these machines, and is high accurate, is less lab work, is cheap compared to Sanger, and the data output is from 150 to 300 base pair. So it's much smaller when compared to Sanger. And the third generation is less accurate compared to the other 2 ones. It's also less lab work, but it's very expensive. For example, in our lab, we were doing a price quotation to sequence 400 plants. It's about 20 million Czech crowns. So it's not feasible for evolutionary studies. But the data output is much easier to analyze because it's from 10,000 to 100,000 base pair. So it's easy to put in this data together and have a single view of the genome that you want to work. And the second thing that you need to choose is the sequencing technology. For example, recovering these molecular regions, we have several types of sequencing technology that we have, but I will be discussing 2 of them today because they were going to be part of the pipeline that I'm developing here. So this 2 sequencing technology is the whole genome sequencing and the RAD-seq. So as the name suggests, whole genome sequencing is the sequencing of the whole genome, but it's not really useful for evolutionary studies because the amount of data generated is never going to be possible to infer a phylogeny using thousands of molecular markers. So, and to assembly and put this whole figure together, you need to have a reference genome. And as you may know, we don't have so many genomes sequenced for most of the organisms, especially for known models. And it's very difficult to analyze because genomes are very big. It's also, you need to have very demanding computer resources, for example, very good cluster to analyze this data. But in whole genome sequencing, you can analyze all taxonomist levels that you want. Do you want to infer a species, phylogenetic tree for species? Do you want for families? Do you want for orders? It doesn't matter. Whole genome sequence is going to work. And the size of the whole genome sequence is variable depending on the sequence technology that you want. But instead, the RAD-seq, that is another technology that we are going to be talking today, is the RAD-seq. And the RAD-seq works a little different. We have two enzymes or one enzyme that is going to cut the DNA randomly in different parts of the DNA. So it's a type of sequencing that we call as genome reduction. So it means that you're not going to have the whole picture of the genome. And in this kind of sequencing, you don't need a reference genome to analyze. These parts that are going to be cut together, they're going to be clustered later. So you don't need really to have a reference. And it's relatively easy to analyze. You still need to have some bioinformatic experience, but it's not as much as the whole genome sequencing. But this type of data set or sequencing technology usually is used for population studies, not really for works for families or orders or in other things. And one problem of the RAD-seq is that the data output that you are going to have, because these enzymes are going to cut a specific place, random places in the genome, but the specific size. So the data output that you're going to have is usually H2 base pair long. So you're going to have problems to assemble this data later. And I'm going to change the topic a little bit, but everything is going to be together in the end. So we are facing real biology, and we know that we are facing very difficult times for science and for humanity in general. So we are losing more species that we can really conserve or understand. And for example, these 4 pictures is by different biomes in Brazil, and most of them, they burned and they lost species and they lost habitats in the last 4 years with our magnificent President Bolsonaro. But if you tell to a person that is not a biologist that, oh, some area is burning, like, yeah, what about it is? So we needed to show some concrete examples of how we are going to losing biodiversity. And in 2015, for example, one of the biggest disasters in Brazil happened, and this was noticing many news around the globe. So it's called Mariana mining disaster, and it happened in Minas Gerais state. And these disasters happened on 15 December 2015. And one friend of mine that worked with sunflowers, she sent a publication for a journal, and you know how the publication works, you send and you get the reviews, and it takes time for publish. And 1 month later, after the disaster, the new species was published. But the problem is that this species is only known for 1 population and 2 vouchers. And it's exactly this area that is flooded. So it was devastating. Probably the species was described and already extincted. So one of the problems that we also have is that we have a lot of species. Yes, this is good. But one problem is that for most of the species, if you assess public databases, you're going to see that less than 5% of the species have at least 1 molecular region sequence. And this is a huge problem, and we can not really understand biodiversity with this last data. But on the other hand, you see that some known organisms, for example, Drosophila and Nicotiana tobacco, has several of the same species sequenced over and over. And before this, we lose all of this biodiversity. It's like clear in our minds that we need to sequence more organisms and also use the data that's already available on the databases. But when you look into NCBI, for example, that is one of the biggest repository for molecular data, you're going to have this picture. So these all are next generation sequencing technologies, and they have different names and they have different strategies. So because we want to include more species and not sequencing the same species using different technologies, one thing that came to our mind is that, is there really a way to sequence, to combine data from different technologies? And in this study, in case of the rat-seq and whole genome sequencing. Because, for example, you could be working the neotropics with some genome that is distributed across the globe, but you only have sample for the neotropics using rat-seq, for example. And you have some colleagues in China that have information for the same genus for the whole genome sequencing. So is there really like a meaningful for you to extract the same sequences in both or increase the taxonomic sample? So this is the reason of the talk, and this is what I was trying to do, is that, is there a way to merge data from different sequencing technologies? So in this case of this project, rat-seq and whole genome sequencing. So if there is a way to do it, how is the best way to do it? And even though this is not a question, is that we don't need to keep this information for us. Let's do a pipeline or tutorial, how other researchers can focus and can do it by themselves. So the first question, is there a way to merge data from different sequence technologies? Yes. And this idea was not mine, was from Natasha Wagner, one of our collaborators in Germany. So for the willows, because we work mainly with willows in our lab. So for the willows, she has a lot of information with rat-seq. But in our lab, we were working with willows and we have whole genome sequence. And we have species that she doesn't have in her data set and vice versa. And at some point, we want to merge this data. And this is the phylogeny, the first phylogeny that I recover, including rat-seq and whole genome sequencing. So this is all the species with merging the two sequencing technologies. So how do I know that it's possible to merge? Because when I inferred the phylogeny, I saw that the whole genome sequencing appears in a clade with the rat-seq. So it's correctly, the species with the whole genome sequence is correct position in the tree. But how is the best way to do it? So there are several ways to do, and I'm going to be showing you two. And the basic idea is that, so we have the rat-seq DNA alignments. And for each alignment, I just put it here, 1, 2, 3, 4. We created a consensus of these alignments. And using this consensus, they are going to act as targets and look where in the whole genome sequence these short fragments are located, and I'm going to extract that information. And there are two ways to do it. So when you receive your sequencing data from the company, not all the reads that we call in next generation are going to be in good quality. So you need to exclude some of the data. And after this exclusion, we call as a clean reads. So we use these clean reads and map directly against to the references. But if you do this, you're going to have a problem, because in the alignment, you can see this is the whole genome sequencing, and this is the DNA information using rat-seq. So you can see that there are some portions in the DNA matrix that there is missing data. And this could cause some bias in the phylogeny. For example, in our tree, the whole genome sequencing always appears as sister group to the RADseq, and could be this missing data biasing this. On the other hand, we have another approach that is called the de novo assembly. So when you send your sequencing or your species to be sequenced, none of the technology that we have is going to sequencing like in a row your DNA. Your DNA needs to be cut in short fragments, but the DNA does not fragment. At some point, you need to merge this assembly to have a whole picture. So if we do this de novo assembly here in black, we are going to face or getting better this problem with especially in the ends. But one problem is that now our whole genome sequencing is much larger as our RADseq, and that also could cause a bias. So in the end, we have to trim these alignments in the end, and this is very hard and very computational demanding. So this is the whole pipeline, bioinformatic pipelines that I created. So we have 2 organisms that we are trying this pipeline, and I downloaded this data from the NCBI, or the repository for molecular data, and I include this data in this pipeline. So I removed the reads or the sequences that did not have a good quality, and then we call them as clean reads, and did 2 types of mapping, using these clean reads directly to the consensus sequence, or I use these clean reads to put the pieces together, what we call de novo assembly, and then we have the mapping of the sequence of the sequences, both RADseq and whole genome sequence together, and did multiple sequencing alignments. And once we have these alignments, we use 3 types of trimming the data for the data have the same size in whole genome sequencing and RADseq for not causing bias on the phylogenies. And lastly, we infer the evolutionary history of these groups. And you may be asking why ficus and why willows. So basically, I would like to try to include as many organisms as possible. So if you go to NCBI, there is a way for you to say, get me all the information that whole genome sequence have for all the organisms and all the information that you have for RADseq. And then I map together to see which organisms have both information in the whole genome and the RADseq. And I was really surprised that there is only 1,000 species with this both information. And then I was selecting the groups based on the number of species, because if you do a phylogeny for a genera that you only have 3 species, it's a kind of bias. You need to have a good taxonomic template to show that your pipeline is working. And then I found around 40 good genera that I could search for it. And after I was looking which studies is at least species level, because as I said before, the RADseq is mostly for population genomics. So you could not really represent, because if you have a phylogeny with several individuals from the same species, and if you put a whole genome sequence in there, of course it's going to be somewhere in the clade. And I only found good data sets for willows and ficus. And recently, Pablo was telling me that there is a good data set for butterflies. So in next steps, I will be trying to see if it's a good data set or not. So these are the results that I have for ficus data set. So this is the phylogeny combined all the RADseq data and the whole genome sequencing. As you can see here in red, this is the data from whole genome sequencing, and in blue, you can see the same information for the same species in RAD-seq. So in all cases, the whole genome sequencing is sister to the RAD-seq. So the pipeline is working and we can combine the data set. Except for one species, Ficus fiasticula, that we were in contact with some specialists in the group in Ficus, and they told me that there was a contamination in the sequencing during this DNA, so we cannot basically use this species because the data is wrong. And for the willows, I still do not have the final results, but some preliminary results that I have, as I showed you in some slides previously. So our whole genome sequencing appears as the sister to the RAD-seq samples. And in this phylogeny is only based on whole genome sequencing, and it clearly matches in all the clades when we combine both information. So the pipeline is working. And so how is the progress so far? Because you may know that I'm leaving soon in 1 week. So for the Ficus data, I already did all the steps of the pipeline, but for the Salix, I'm still doing this type of trimmings that I was saying before. There is 3 steps of cleanings, and I would like to include another animal in the pipeline, perhaps the butterflies. And the reason why I want to include another animal is that because in animals, usually you don't have so complicated genomes as the plants. So they don't have so much hybridization, introgression, and so on. So, and this is not the question, but one of the goals is that I will be creating a GitHub. So this is how bioinformaticians communicate in science. So in GitHub, if you go there, you're gonna see many packaged softwares and so on that you can analyze your data. So I will be creating a step-by-step way to how to analyze and all the programs that you need to perform these merging sequencing technologies. So that's all. Thank you.
Thank you. Anybody else on the topic?
Yeah.
I would be interested in the 2 methods. How much are each of them used in the community at the moment? Is RAD-seq more used, or is whole genome sequencing more?
It's usually more RAD-seq because the whole genome sequencing is very expensive. It's very difficult to analyze. It's not usable because in the end there are several repetitive elements and some parts of the genome that you cannot use. So RAD-seq is the most used nowadays. Yeah.
Yeah, I wonder if you get this like expected relationships between the whole genome sequencing and the RAD-seq, but do you know if it affects the branch lengths of the?
No, it does not affect it. You cannot, because I put it cladograms, right? I transform it. So no, you cannot see really branch length different.
Okay.
Yeah. Mm-hmm.
You were saying that RAD-seq is more widely used for population genomics.
Yes.
Not for phylogeny.
Mm-hmm.
What's the limitation for using RAD-seq for phylogeny?
Because they are very short, usually. They are only 8 base pairs. And the people, when you have some information about the genome of the plants or the organisms that you are working, you prefer to design probes and work with non-regions, because in the RAD-seq it's random. So you don't really know where this species is from. So if you want to study some evolution of some genes in colors and wings and so on, you don't really know about it.
So, but you were saying that whole genome sequencing is not used. So what is being used for phylogeny now?
For phylogeny, mostly if you have some information of the genome, use a target enrichment. So basically the target, I didn't say here because this is not the focus of this merging sequence technology, the pipeline that I'm creating, but you kind of design probes or complementary sequence to the regions that is known. But for designing this complementary sequence, you need to have some information of the genome. And for most of groups, we don't have this information. So this is why the people are using sometimes RAD-seq for species level, but it's not common to see.
Okay, there are two questions in online,
Yeah.
Vic and Martin.
Mhm.
Can they speak? Can we hear them?
Okay, I can maybe start because I can comment on the question probably Leo asked now, why we don't use always RADseek, for example. So I can comment on the situation in Willows. So basically RADseek is sometimes, the data are too variable. Yeah, so for example, in Willows, we would not be able to merge data from two different subgenera because simply the data would not overlap.
You can't build a common
Exactly.
alignment.
Mhm.
Okay, thank you. And Vic? I don't know. Tell me, Vic, you can ask your question. Like,
Can you hear me?
yes,
Yeah.
cool.
Okay, so I was asking how many markers these last phylogenies that you were building, that you were showing in your results. These are basically in the end rad tags, whether they come from whole genome sequencing or RADseek. How many of them did you use to build them?
So depends on the organism groups that you are using. So for the Willows, we had 23,000 markers or molecular regions. And for the ficus data set, we have 500. And for the butterflies, I checked earlier, is 100,000.
Okay, and in quick response to Martin's comments, is it out of all the RADseek data, you should still be able to find something in common if you're looking at different species or even genera, right?
Yes, yes. Yeah. Is that a question or?
Yeah, thank you.
Yeah, definitely, because when you have whole genome sequencing, you have the information for the whole genome. So you should be able to find these small regions in the RADseek in the whole genome sequencing. But perhaps it's not true when you're trying to merge with another sequence technology. For example, the target arrangement that we were discussing before is just a representation of some genes or some other regions. So it's a genome reduction. So if you try to find the RADseek that is already reduction in something that is already reduction, perhaps you cannot merge them.
Yeah. Real nice presentation.
Thank you.
Okay, thank you.